It's like 402. It's like 402. Cool. Squeaky spot. All right. Well, welcome to the second seminar of the evening. There it is. So welcome out to the second seminar of the year. We have Luke Bauer, postdoc in our lab. He's here studying environmental flows for stream fish, but he'll be presenting work that he just finished from his dissertation at Texas A&M. So Luke got his BS at just down the street, Erskine College, Southeastern Louisiana University for his master's, and then here you see Texas A&M University for PhD. And he landed in Clemson just a couple months ago. So, yeah. So Luke has been all over the world, as you'll see, sampling stream fishes, and his work in general focuses on ecology and evolution of freshwater stream fish. And like I said, he's here working on the effects of like in-stream flows on stream fishes. So continuing on with that. So welcome, Luke. Look forward to hearing what you have to say.
Thank you, Brandon. So thank you everyone for coming. I thought this would be a good opportunity for me to introduce myself to the department and everyone to get to kind of know what kind of research I do and where I'm coming from. So today I'm really, as you said, I'm going to focus on my dissertation, and then I'm going to talk just briefly about what I'm going to do here at Clemson. So my dissertation was really inspired by MacArthur, and so he had stated that the goal of all science should be to search for these general patterns or rules. And this can be applied to physics, chemistry, and community ecology. And so at the time I was reading a lot about community assembly processes. And these are the processes that shape the identity and abundance of species within ecological communities. And so I wanted to aim my dissertation at trying to find general patterns of these community assembly processes. But anyone that's studied ecology knows that it's not as simple as that. And it's driven people like Lawton to say that community ecology is a mess. And that, it's impossible to find these general rules or general patterns. And there's just too much variation and complexity in these ecosystems. But this may not be completely out of reach. McGill and others have suggested that functional traits may be a way of doing this. So functional traits are really anything that a species has that determines where they can live and who they can interact with, or interact with other species. An example of this would be, the bill shape of birds. Hummingbirds have this really narrow, small bill that it can use to get down on flowers and get nectar, versus, a great blue heron that has this more of a spearing type of bill for catching frogs and fish, but not so good at getting nectar out of flowers. And so you can use these functional traits to kind of get an estimate of what the trophic niche of these organisms are. We know the difference in their diet just based on that bill shape alone. And functional traits are really good at mediating the interactions between the environment and the species. And they also are really good because they can represent different niche dimensions. So you can have, for example, the bill shape here representing a trophic niche dimension, or you can have, for maybe wing shape or some other characteristics that represent a habitat niche dimension. And so this really improves our mechanistic understanding of the ecology and the species' environmental interactions. And the best thing about functional traits is it's not constrained by taxonomy, so rather than just looking at the taxonomic makeup, you can look at the traits you can look across regions. And so you can look at this across many different places. So today I'm going to be talking and trying to find these general patterns in a few ways, but I'm only going to be talking about 2 chapters today. One chapter focused on convergence, and another chapter focused on the functional and phylogenetic structure of these stream fish. And this third chapter, it was just recently published, so if you want to read about that, it was published in Ecography. So my first chapter looks at convergent evolution, and I define that as the phenotypic similarity among 2 or more phylogenetically divergent taxa, and so or species. So this is not a mechanistic definition, and oftentimes people will see lots of convergent evolution, like the rift lake cichlids in Africa, and they assume that this has to do with adaptation to some selective pressure, and that's normally how we think of convergent evolution. But that's not always the case. You can get convergent patterns coming up from just constraints. So if fish can only exist in 5 different ways, if you repeat this over and over again in different areas, eventually you're going to end up with convergent forms based on just constraints alone. And these constraints can be mechanical, it can be evolutionary, or just developmental constraints. And they can also arise from just purely random. This guy named Staten, he just created these random models and created random taxa, and he was able to produce convergent forms purely by random. And so this makes finding the mechanistic background of convergent evolution a little tricky. Another thing that makes convergent evolution pretty hard to study is that it's not perfect. This is like a classic textbook example where you have this shark and a dolphin. Another one's a bat and a bird, but you can tell bat and bird are not that similar. A dolphin and a shark don't look that similar. So what is similar enough to be called convergence? And that's another problem. How similar do you need to be convergent? And so convergent really lies on this gradient. It's not yes or no. You can be very convergent, you can be slightly convergent. And so this is also a difficulty, but luckily there's some methods that can deal with this question of similarity. How similar are you similar enough? And I'll talk about that in a little bit. Another problem is this many-to-one, a relationship between form and function, or many-to-many and many-to-one. So here, for example, gape size in fish is often constrains what they can eat. So here I have a large mouth and a large mouth of a goby. We could probably eat that huge long-armed shrimp that my lab mate could not fit in his mouth. So it's a trophic trait. But it can also be associated with breeding, such as like these kissing gouramis, or it can be associated with habitat as well. So this is a sucker mouth board-breeded catfish. And we have these sucker mouths that they use to scrape rocks and algae off of rocks. But it can also be used as like a sucker. They hold themselves on rocks in these really high-flow environments, so they can persist in these high-flow environments. And that way, it's not only a habitat trait, but it's also a trophic trait. So it also muddies the water for studying convergence. And so generally in studies, when they look at convergence, they're looking at pretty limited scales. So either phylogenetically, they're looking within a single genus, like these amphipods, or the famous examples with Losos and the anolis lizards in the Caribbean. They're really looking at within a single genus. Not only is it phylogenetically limited, but oftentimes regionally limited. They're looking at one or two areas, or even within a single area. And when studies do, and this is a study by Moen, when they do have this large geographic scale and phylogenetic scale, they often use very broad characteristics like large, medium, and small, long-legged or small-legged, things like that. So I was hoping to do a convergence study looking at a large geographic phylogenetic scales and using more fine traits. As I said before, oftentimes convergent evolution is associated with adaptation and selective pressure. And if that's really true, this brings up the question of if the habitats have stronger selective pressure, would this cause the degree of convergence to be stronger? Would there be stronger convergence? And I would like to think of this in an adaptive landscape way. So if you have traits in adaptive landscape, you have this environment, if there's a strong selective pressure, it should cause these really tall, narrow peaks in the adaptive landscape. So you should have a very narrow morphological range that can exist in this habitat, and you should have stronger convergence. But if you have weak selective pressure, those peaks of the landscape are very broad. So you can have many morphological forms existing in that habitat, and they're more than likely going to be weaker convergence. And so I wanted to look at this question, and fish is good to study these questions because they live in areas with high selective pressures, like water velocity. So in my previous chapter, I saw that water velocity really restricts traits. So here, this is body depth on the y, water velocity on the x. And you can see once you hit about 0.2 meters per second, you lose a lot of that body depth variation, and you have this restriction of the traits. So water is acting as this environmental filter. And that's because when you have these deep-bodied fish like this sunfish, they cannot exist in these high flow areas without expending a lot of energy because they're not very hydrodynamic, like these fusiform fishes like this loach. And you can see this in multiple other traits. I saw this in various traits. We have this restriction of morphological variation as you increase water velocity. Another habitat variable that does this is substrate. So here's a measure of substrate complexity. And you can see, so here, complexity just being like things like woody debris, aquatic vegetation, some kind of structure in the water versus just pure sand. So you have structure over here and then pretty much pure sand over here. And you can see there's this huge restriction in the morphology that can exist in these habitats. And of course, in ecology, there's always one exception, like that little dot over there. And that's this guy. And so he's a geminiform Genera-Ranthichthys, and he's a sand-bearing electric fish, so he produces a weekly electric fish. And so he kind of screws everything up for me as far as clean results. So the hypothesis for this study, my first one was that I would see convergence in these very similar habitats, and that should be found across all these zoogeographic regions. And also species from habitat groupings with high velocity or non-structured substrates should have higher degrees of convergence and lower functional riches. And so to accomplish this, I sampled in 5 different places along a latitudinal and longitudinal gradient. So I sampled in the United States, Belize, Brazil, Benin, and Cambodia. And within the United States, I sampled Texas, South Carolina, and New Jersey. And so each of those sites, I was looking at 3 different scales. So the microhabitat, a stream reach, and then the whole zoogeographic area. And every time I went to a place, I tried to sample at least 6 streams, and they had to be similar. So I was trying to restrict the confounding variables and find very, very similar streams all over the place. So here's some pictures of those streams. They had to be sandy bottom, they had to be low gradient, they had to be forested and pristine. And here's just a picture of Belize. And so this is a pretty hard thing. It's probably the hardest part of my project, was actually finding these sites wherever I went. And not only that, we found, it's difficult to sample with all kinds of Latin critters that we found out and around. But I think the most dangerous thing we ran into was the locals around East Texas. This guy stopped by to show us his prize zucchinis. And then we also had some female visiting students from Turkey and Brazil, and he was trying to get them to go back and drink moonshine with him, which luckily they did not. And I also ran into going crazy in the field. This is one of my undergrads was helping me in Belize. His hands got swollen really purple while we were in the jungle. We're not sure why, just the fingertips, but luckily he didn't lose any of his fingers. And of course, we had equipment break down wherever we went. I don't know why, I have a curse with trucks. They break down wherever I go. And I've been left on the side of the jungle to get your bus home eventually. So once I make it to these sites, I sample roughly about 200 to 500 meters. And then for a stream reach, and then within any of those stream reach, I sample about 40 to 70 microhabitats. So roughly probably an average around 60. And so a microhabitat was this heterogeneous habitat type. So if this is a stream, we'd come and sample. We'd see that this is a sandy pool. We'd sample a few meters around that little area. We'd collect those fish we put them in a bag, flag where we got the fish, and come back and measure the substrate flow and depth of that microhabitat. So we know where we got every individual fish and what habitat they were associated with. And I did that for the 200 to 500 meters. And then for each of those species, I measured 30 functional traits. I standardized them, and then I grouped them up into trait groups. So I had traits associated with habitat use, traits associated with diet, and I got life history characteristics from the literature. And then I combined all those into an all traits category. I also got a phylogenetic tree from the literature, from a recently published tree that's really helped me out. And then I was able to calculate phylogenetic signal and also do a couple other analyses using that tree. And so for this convergence project, I needed to group fish into habitat groupings. So to do this, I did an RQL analysis and I took those axes close to them. And I ended up with 7 different groups. So an example of that would be fish that are midwater that tend to be in microhabitats with low water velocity or high stream and high stream structure. And so I had 7 of those types of grouping, and then I sort of arbitrarily broke species into species that have low affinity or high affinity water flow, and the same thing with structured substrates. So to actually find if species are convergent, I use something called a tangogram. So this looks really confusing at the beginning, but here is a phylogeny, and here's a dendrogram or morphology, so this is the traits that I used. And so this is not mine, this is an example of one. So convergence are shown by these dark pink, these thick pink lines. If you're phylogenetically distant related, but morphologically similar, that would indicate convergence. So that's how I went through and found convergence through my data. And then to see if this conversion was significant, and also to get the degree of convergence, I used state and Cs. And so this is just characterized as the degree of divergence from extant taxa to the ancestral state. So you can kind of see how much of the morphological distance was reduced throughout this evolutionary history. And this ranges from -1 to 1, and 1 being that they are perfectly convergent. And so here's just a summary of some of the results. I got roughly about 230 species in total, and then this is species for the country. So looking at my first hypothesis was that I'd find convergence in these similar habitat types. I did see that. I saw about 70 out of 197 species. So that's about 36% of the species were convergent with at least one other species. And this is an overwhelming slide, but all I wanted to show is that this is my tangogram, so the phylogeny and my morphological tree, and then these are the habitat groups colored. And one interesting pattern you can see is that not all of the groups converge into one area, that some of them are spread out. And this suggests that there's many forms for some single function. So this many-to-one relationship. And here's just a better view of my convergent taxa and how they're spaced out on the phylogeny. And once again, they're colored by their habitat grouping. And a closer up at some of these, just a few examples of those, because there are 70. So here are some species that are found in fast-flowing water habitats that are generally structured. So all these species tend to have this fusiform body shape, this really hydrodynamic body shape. They all that allows them to persist in these high-flow habitats with expending little energy. There are these large pectoral fins that they can use to create negative lift to push them against the substrate so they can hold their position. And all of these species, or not all of these, so 3 of these are all in different orders, and these are in 2 different families. So they're pretty phylogenetically divergent, yet they look very, very similar. Probably my favorite example, and one I did not expect to find, are these 2. This is a leaping barb, and the silvery one is a leaping barb, and then you have a hatchet fish up there. And they live in similar habitats, but I think the neatest conversion is actually their predator avoidance. So what they do is they have these, you can kind of see the top here, these huge pectoral fins, and like flying fish, they just use those to launch themselves out of the water when a predator comes, and so they can like fly out of the water. And this guy had a beautiful blue spot on his head. And it makes them very, very hard to catch when they're jumping out like that. And so we also had examples of convergence with these species associated with low water velocity and high structure. So here we have from all different families. And they tend to have these deep bodies and large dorsal and anal fins that really optimize them for these structured habitats where we can make these quick, sharp turns. We also had examples of convergence with predatory fish, and there was many examples of that. And one thing I really liked, I was surprised by, and I didn't really notice before I started this project, is fish that were associated with only pure sand tend to have these clear body colors. They all tend to be pretty clear. No matter if they were catfish, this is a darter, this is Amicryptobenine, even those gymnotiforms, electric fish, all had these really clear body types, which is something I couldn't test, but something that I saw was pretty neat. So looking at my next hypothesis, I wanted to see if these high water velocity habitats would have higher degrees of convergence, these C1 values, and lower functional richness. And so what I did see that, I saw that in a high water velocity, you tend to have much higher C1 values, and you tend to have much lower functional richness, basically the area and morphospace you take up. And then for I was looking at the same thing for structured substrates. So I expected that areas with high or low structural complexity would have high function or C1 values, and it tended to have high C1 values, and also very low functional richness. So this really shows that these water velocity and structure complexity are strong environmental filters for fish. And they tend to restrict the traits that can occupy these certain habitats. And again, if you think of that in that adaptive plane, you think if you have strong selective pressure, you have these very narrow peaks. So you can only fit a few morphological types along in this adaptive landscape versus low selective pressure where you have a really broad area that you can occupy. And so as I said, these high selective pressures tend to lessen the functional richness and increase the degree of convergence. So I did see a lot of widespread convergence in these certain microhabitats. I also didn't show this, but I saw a lot of phylogenetic signal in these traits, and this suggests that there is phylogenetic niche conservatism or some kind of evolutionary constraint acting upon these assemblages as well. And that water vorticity and structural complexity tend to reduce this functional richness, increase the degree of convergence. And so this shows that convergent niches seem to be fairly predictable among these streamfish, with species from widely divergent lineages having evolved in response to similar adaptive peaks defined by similar selective pressure. And this suggests that there is some kind of non-random or deterministic mechanisms that are influencing these assemblages. And so it's not just all purely random. So now I'm going to switch to something completely different. So this part of my dissertation was really focused on those community assembly processes. And those are the processes that structure the identity and the abundance of species within a community. And I was focused on two, so environmental filtering and limiting similarity. So environmental filtering is just the idea that the environment prevents certain species from being able to successfully inhabit a given habitat. So a really broad example would be, here these deserts tend to be hot and dry, and so that would prevent trees from a cloud forest from ever being able to live here because they're environmentally filtered out before they can get there. And limiting similarity is the idea that species will avoid competition by segregating along a niche axis. So again, that hummingbird and great blue herring, they may live in the same habitat, but they're not competing because they've segregated against a trophic niche or in some other way. And so when looking at these mechanisms, there's no really consensus regarding which one's more important. So we don't know which one's relatively more important than the other, and there's no really general proposed rules for these processes. So, and most of these studies have focused on plants and microbes, and not really that many large-scale studies. Normally they do this in one stream or one area. And another problem is a lot of these studies use very different methods, so it's hard to compare across studies to find any general patterns because their methods are so different. So my idea is to try to look at these assembly processes using functional and phylogenetic methods, using similar methods at a large geographic scale, using vertebrate fish. And so to do this, these methods, so if you think of this as a theoretical fish community or regional species pool, if you have an environmental filtering, the environment should restrict these certain species from being able to persist, so you should end up with a local community with very similar traits. Like community A, they are very, very similar. But if you've got limiting similarity, then they're trying to segregate in these niches, so they're trying to not be similar. So what you should end up with is over-dispersion, a community that is very, very functionally different. And so that's the idea of, so you can use these trait patterns to kind of estimate which mechanism is acting on these assemblages. And scale is really important for these types of studies too. So at the regional scale, and at two different scales, you have different mechanisms that are possibly influencing the communities. At the regional scale, it could be speciation, extinction, dispersal, and also abiotic factors. But at the local scale, you move more on to species interactions, and other environmental factors. So another thing, I was looking at the stress dominance hypothesis, and so one thing that this brings up is that functional diversity should be reduced as environmental stress and stabilizing selection increases. Unfortunately, there's been pretty inconsistent support, so I thought I would try to test this with fish, because they are great, and we saw that water velocity and structural complexity tend to have this large influence and this stressful influence on these fish. And so one of my objectives for the study was to evaluate the similarity of functional and phylogenetic dispersion patterns across these microhabitats, or at the microhabitat and regional reach scale. And one of my hypotheses, or really two of my hypotheses for this objective, was that environmental filtering should be the most important process, regardless of scale, and at all these different places. And that over-dispersion should really, so this is limiting similarity, should really be most important in the tropics, where supposedly competition is the highest. So I also wanted to test the relationship between these environmental gradients and the functional metrics to see if these functional diversities would decline with increasing water velocity, decreasing water depth, and non-structured substrates. So again, I'm looking at all these different places around the world. I'm looking at 2 different scales. So I'm doing stream reach scale, which is at 2 to 500 meters, and then the microhabitat scale, which is at a few meter sampling point. And I'm using different trait groupings. So I again cluster them by habitat, diet, and then a combination of traits. And I did this because if you're expecting the environment to influence your traits, it should influence the habitat-associated traits most. And with the separation of the niches, you'd expect that they're going to be trophically separated, and they should focus on the diet traits. And the combination should give an overall picture. So I'm using 3 different metrics: Rao's quadratic entropy, functional richness, and then mean nearest neighbor distance. And then I also want to look at phylogenetic dispersion. So I'm using the mean pairwise distance. So that's just the mean phylogenetic distance between every single individual and the average of that for that assemblage. And then mean pairwise, mean nearest taxon distance, which is the minimum distance between a species and all the species in that assemblage. And so to see if something's really got, over or under dispersed, or if there's environmental filtering or limiting similarity, you need to compare this to a null model. And so we calculate these standardized effect size. So we compare our observed value to what you get from like a random model, and to see if things are under dispersed or over dispersed compared to random. And so negative values would indicate under dispersion, which is environmental filtering like A, and positive values should over dispersion like B. And so to create these models, what I did is, if this is my species, my regional species pool, this is my local species pool, I would randomly select species from this regional species pool to create random communities, and then compare that to my observed values. And I ran 2 different selection algorithms for this: independent swap and taxon label. So the results from this, I wanted to evaluate the similarity of these phylogenetic and functional dispersion patterns at the regional and microhabitat scale. And this is a rather overwhelming slide, I know. So on the columns of my different countries, so Belize, Benin, Brazil, and Cambodia, these are my different functional metrics. And then each individual column are my habitat groupings. And so black is under dispersion, dark gray is no different from random, and then light gray, the little bit you can see, is over dispersion. And so what you can see as a general pattern is that the black is way more, you have more under dispersion than you do over dispersion. But there's a lot of just pure randomness in these assemblages. We do get some significant, quite a bit of over dispersion in Belize with the trophic, so that suggests that they're trophically separating in these assemblages, and then Cambodia. And I'll talk just a second why those may just only be those 2 countries. So we saw a lot more under dispersion than over dispersion, and this is looking at just the microhabitats, but this is also true at the reach scale. So we saw over dispersion in Cambodia and Belize, but not really where I expected. I expected to see a lot in Brazil and as well as Benin. But so one thing maybe there's not limiting resources in these habitats, so you need limiting resources for competitive exclusion. There may be enough disturbance in these small headwater streams to prevent species from ever being able to compete, because they're keeping their population numbers low enough that they're not competing. And another reason, probably the reason I think for at least Cambodia, is that the regional species pool size is large. I had a lot more species in Cambodia. I was sampling slightly larger streams, and there was a lot more species in there. So if you have more species in all these habitats, it's more likelihood that they'll be competing than when you have less species. I see a lot of these non-significant results, and this suggests there may be some stochastic factors influencing, so you have more stochastic processes going on. Also, this may be driven by contrasting patterns of these plates. So if you have over dispersion and under dispersion working together at the same time, you'll get this net neutral pattern. And so to give you an idea of what's happening there, so this is a graph again. You have environmental filtering, and you have a limiting similarity, so over dispersion and under dispersion. You can have where you have environmental filtering happening first. So here, these species are really coming from this area of morphospace. There's a huge restriction due to environmental filtering, yet these species have different niches and they feed differently. So the colors represent different niche types. So you can have this over dispersion within a under dispersion thing where you get basically net neutral and you get no results at all. And so to try to counteract that, I tried to subset the null models into habitat types to try to account for any previous environmental filtering. Also, I may have selected traits that are just not that great for showing competitive exclusion or environmental filtering. So, the traits you choose are also really important. Another problem with that is that many-to-one mapping of form and function. So again, that mouth, it can be associated with diet or with environment. So you can also have these traits that don't really show any good patterns. And then another reason you may have got a lot of these neutral or non-significant results is because these are really small headwater streams, and there may be frequent hydrological disturbances. Anytime there's a strong rainfall, these streams are going to flood and wash out pretty easily. And so that may have caused these non-significant patterns. And so this is really looking at my phylogenetic dispersion results. So here, again, the black is underdispersion, which means that these species are more related than random, so they're more closely related than random. And then overdispersion is the light gray, so they're more divergent or not related than expected by random. And then I got a lot more of this just random, non-significant results. However, these phylogenetic dispersion metrics, you have to interpret them with concern, and really they've been shown not to show any of these community assembly processes. So they're better used to try and look in a different way. And I came up with I think is a fairly novel approach, which is very simple, is trying to graph the functional diversity with the phylogenetic diversity and try to get at what's happening. So again, here is my phylogenetic metric, so dispersion metric, and here's my functional dispersion metric. Negative numbers means they're underdispersed. Positive numbers means they're overdispersed. So if you put these into quadrats, you can kind of assume what's happening in that assemblage. So for A, you have species that are phylogenetically related, but morphologically different. So that would suggest that these are divergent species. This assemblage is made up of divergent species. Whereas B, you have these related species with similar traits. So that's niche conservatism, or just similar species occupying these other way around. Sorry, these niche segregations. So these are overdispersed. So they're not related with not similar traits. So they're just unrelated species. Whereas C is stabilizing selection over time. So you have these species that are unrelated or very related and have very similar traits. And finally, D, where you are morphologically similar but phylogenetically distantly related, suggests that these species are convergent. So looking at my other hypothesis, I thought that these functional metrics would decline with increasing water velocity and also decline as things get shallow and non-structured substrates. And so what I did see is that functional richness did reduce with water velocity and with depth. And so this suggests that water velocity is not only a strong filter, but it also reduces that functional richness. And on depth, this may be due to just the size of the species. As you get shallower, you're not going to have bigger species, maybe predators, forcing those some morphotypes out. And then I also saw with structural complexity that two of my other functional metrics decreased in non-structural habitats. So this really shows that environmental filtering and to a lesser extent species interaction influences these assemblages around the world, but really that environmental filtering is a pretty universally important assemblage process. And that as you increase the environmental stresses, you tend to increase or decrease the functional richness or the functional diversity of these habitats. And then I found a really high incidence of functional trait dispersion coupled with phylogenetic underdispersion. So this really suggests that there's still a lot of phylogenetic conservation or stabilizing selection, some kind of evolutionary constraint. And so not only do you see lots of convergence, but you also see a lot of this evolutionary history, this evolutionary constraint. So just kind of a summary of my dissertation. I was looking at all these geographic regions. I saw this pretty predictable response to these environmental filters, which hopefully has led to this prevalence and convergence. Stream and water velocity tend to be strong influences on these fish assemblages and really important. So that has conservation applications. So if you're taking away these structured habitats such as the riparian zone, you're cutting that away, you don't have any more root banks or wood in the water, you're really going to reduce the functional richness of these streams. Also, heterogeneity in flow is very, very important. So if you're altering these flows, you're also going to influence the functional diversity of these streams. And one thing I found extremely annoying, and I wanted to get on a soapbox for just a moment, is the lack of natural history for a lot of these species. Even in the United States, what I thought would be common species, there's a lot that is not known. And it's really preventing me from doing a lot of the work that I wanted to do. And unfortunately, I don't think there's that much funding for these natural history studies, and I kind of wanted to promote getting more of these natural history studies going, especially in some of these countries that have, they don't have that much resources to do expensive science. They can easily do a lot of these cheap natural history projects. Another problem is that there's not that much information on these trait performance curves, or like how traits really respond to these different environments. So you need more lab experiments testing the trait performance relationships. And so I was going to briefly talk about what I'm going to be doing here at Clemson. So I'm focused here on in-stream flows, and in-stream flows have always been thought of as the master variable, the most important variable for structuring these aquatic ecosystems. And so maintaining these natural flows is a huge focus right now, and so that we're trying to protect maintain these natural flows to keep these healthy aquatic ecosystems. Now, but unfortunately, there's a lot of anthropogenic alterations. This is probably the most dire threat to these aquatic ecosystems. And so this has led to the field of environmental flows, which is just studying the quantity, timing, and quality of these flows required to sustain freshwater biodiversity. And so the goal of the project I'm going to be working on, and one thing is that a lot of these studies are focusing on larger rivers, where on this study we're going to be focusing more on these small streams, and not that much work has been done on small streams. So the goal of this project is using these biological criteria to try to come up with stream flow standards for the state of South Carolina. And we're also going to be trying to come up with quality of conservation, or put a value on these in-stream flows for these little freshwater streams. So this is a pretty collaborative project with Texas A&M, SDNR, and the Nature Conservancy, as well as RTI and International. And really my role right now is trying to quantify these relationships between these flow variables and the biotic responses. So I'm really trying to find a threshold. If you have so much percentage change in flow, what happens to the community? And try to come up with a number so we see, hopefully we'll find these thresholds of huge change. So SDNR sampled all over South Carolina. I think we have roughly about 400 sites that we'll be using. And right now I'm just waiting on RTI's hydrological flow models. And hopefully I'll be able to present this work in a future date and give a whole presentation based on this. So one thing I wanted to acknowledge, a lot of people, since this is a pretty field work heavy, I had a lot of help during this. My advisor, Kirk Winemiller, really was a great advisor and helped me out with a lot of this, especially helping me get connections to sample all over the place. And my committee members would give me great feedback on this dissertation, especially with my field help. These two guys at the top here, they went to a lot of these field sites with me and literally bled for me many times. So I really am happy for them. And then these two here, Dr. Alphonse and Jansen, they went out of their way to help me a lot, especially Dr. Jansen. He was an incredible help in Brazil. I wouldn't have been able to accomplish this. He even drove 45 minutes one morning and dropped everything to get me a vehicle because one of mine had broken down. So he was a huge help. I wouldn't have been able to do it without him. And of my funding, the NSF Dissertation Improvement Grant, and then the fellowships that I had at Texas A&M. And I'll leave this up and take any questions.
Questions for Luke?
curious when you're talking about the over-dispersion and under-dispersion, one of your kind of solutions was trait groupings. I was wondering what you would expect to see if you grouped all of your fish by either predators or prey and kind of redid that just to see if there would be more clear results. Kind of thinking that maybe prey would be more dependent directly on the environment versus predators are more indirectly dependent.
Yeah, so, yeah, that would be, I don't know if it would add complexity or add simplicity to that, because some of those predators are really habitat specific and some of them not. Some of them are like wolffish, they're just sit and wait predators, so they're always found in the same habitat, so it doesn't really matter about the prey fish at all. They're just sitting around waiting for something to. So I don't know if grouping it by prey and predators would, it may influence the results, but I'm not sure how it would do that. Because like a bass here kind of cruises around and does its thing looking for things, but there's a lot of sit and wait predators as well. If that answers your question.
When you mentioned the high, low, median of substrate, based on what?
So, those I characterize the fish's preference for structure and complexity. So what I did is I collected all these fish and basically got the abundance of fish in structural complexity versus not structural complex habitats, and found the proportion of those fish. So if they're always found in structural complexity, they would get a value of 1. If they're never found in structural complexity, they're 0. And I kind of gave them a habitat preference and then divided them based on that. If that explains. Those are pretty arbitrary groupings. I just divided it into thirds as well. Where the flow metric, I divided more on what I'd seen. Like at these flow, I tended to see these breaks in these communities. So, yeah.
All the same period of the years?
Yeah, so I always did either dry or summer seasons, depending on where I was. I tried to avoid any, you can't really, so a lot of these streams you can't sample in the wet season because it's just too flooded, too much rain. So I always went through when the water was about the mean flow or just below the mean flow. I always had to have waitable, I need to walk through the streams.
So you're talking about some of your results, you didn't find patterns, evidence of like competitive things structuring communities. Because that could be because like streams are too high in disturbance, right? Not enough to let them reach equilibrium to where they reach competition. Do you think that if you did this like in more stable systems, like in lake systems, this would shake out, or you could do that?
Yeah, that would probably shake out in lake systems. Also, I was wondering if I did it instead of doing a low gradient system, doing it in a high gradient where they're pretty, like the flow is pretty standard throughout the year. I picked these low gradient streams because they're basic convenience. They're easy to find similar streams no matter where I went. But now I kind of have that regret. If I had found more stable, even larger rivers, I think I would have had, they're more stable, and I think I would have found much better results in larger rivers rather than these tiny, flashy streams. So, hindsight.
Always another question. Any other questions for Luke? All right, appreciate it.